I'm going to present my recent analysis, data mining analysis on host-drug interaction in drug-induced liver injury. In the drug-induced liver injury, most of our effort dedicated to find out drug property associated with hepatotoxicity and also host factor, mainly genetic factor associated with the risks. However, I thought that either factor, drug or host alone, cannot fully explain individual drug-induced liver injury risk. So I thought there is specific interplay between drug property and the host factor. This is a kind of preliminary exercise for us to find out what kind of hidden pattern we can see when we are using large database, basically mining the data. We identified several group of drugs which are associated with gender age biased reporting. For example, drugs which more significantly reported among women, men, and also young women, because I'm interested in whether or not the pre or postmenopausal women actually influencing the susceptibility. Okay? And also we analyzed the younger generation and the older generation, age generation biased reporting, regardless of the gender. And we found out that women biased reporting associated with the drug which has potential to generate reactive metabolite in the liver, and also more hepatocellular toxic, like the causing mitochondrial the toxicity, or the transporter inhibition. We thought that women biased the biology. For example, and women usually have the increased activity of ZIP enzymes. The women biased attribute might interact with those drugs which generate reactive metabolite in the liver, and both together actually increase the risk of drug-induced liver injury. The other aspect is women are associated with enhanced immune response to any type of injury. With that diathesis, the women may be the more susceptible to liver injury once the hepatocellular injury is initiated. So there might be some potential interesting specific interplay between drug property and age gender related attribute. The WHO, the database accumulate more than 8 million reported cases from throughout the world. Most of the report came from North America and Europe, but pretty much I think it was 80 or almost 100 countries in the world, various sources. So the people think that spontaneous adverse event reporting system, the data quality is not so good. We cannot get any relevant information. But I believe that's the very good resource for type of theory generation. This is a very preliminary effort we generated a couple of hypotheses which we are planning to test using a large electronic medical record system like a VA medical record system or Kaiser. So once we validate what we observed in this analysis, we can start building more the knowledge related to specific drug and host interaction, and that will eventually help us to better stratify the patient in terms of DIL risks, specifically for agent we are using, and that way we can personalize our preventive strategy in the future. We cannot expect to have larger data set. When I say large electronic medical record system, we are talking about maybe a couple of million, but still that data set should have case under control and the well-established the chronological relationship between the drug and event. So it's just a different data set and a different approach.